In Chapter 6, you took a look at the cell and an overview of all of the organelles and pieces and parts involved with the cell. In Chapter 7, we are going to focus on one of those components. We are going to take a look at the cell membrane, or you may hear me refer to it as the plasma membrane. The cell membrane is the outer boundary that's on the outer side of the cell, both in plant, animal, fungi, prokaryotic, all types of cells have a cell membrane. And one of its main jobs is to keep the inside of the cell, so all of the goop, cytoplasm, organelles, stuff that's on the inside, to keep that stuff separate from the external environment. Another job of the cell membrane is the fact that it's selectively permeable. It allows some substances to cross the cell membrane more easily than others, and we're going to take a look at how it actually does that later on. The focus of this lesson is to look at the structure of the cell membrane. Now, as you know, the cell membrane is made up of phospholipids, and just jumping ahead here, we know the phospholipid is made up of a phosphate group attached to a glycerol, attached to two fatty acids. And we've also discussed the fact that these molecules have both hydrophilic and hydrophobic properties. The phosphate head is hydrophilic, so as you can see there's little water molecules attracted to this region. And the hydrophobic areas, those fatty acid chains, are hidden away from water, so they face inside of the membrane. Now, another term that we did not learn that we're going to introduce today is the fact that these phospholipids, because they have both hydrophilic and hydrophobic properties, we call them amphipathic. Now let's examine and kind of dissect this word quickly. The beginning part of this, the prefix of the word amphipathic, you have seen that somewhere before. We're talking about amphibians, right? Amphibians kind of lead a double life, and that's what that prefix kind of means, double life, right? Amphibians live both in water and out of water on the land. So amphi kind of means double life. So phospholipids are both hydrophilic and hydrophobic, so they kind of live that double life. Now, in addition to phospholipids in the cell membrane can also have proteins either embedded inside of it or stuck to the surface of it. Those proteins can also have amphipathic properties. They can have both hydrophilic and hydrophobic regions. Now, the model that we're going to be studying is called the fluid mosaic model, and we will break that down and look at what that actually means. Now, looking at an overview of a very simplified phospholipid bilayer, you can see that it's made up of two layers. That's where the bilayer word comes from. So two layers of phospholipids, and notice that the hydrophobic regions are on the inside and the hydrophilic regions are facing the outside. Both the external environment of the cell and the internal environment of the cell is aqueous. In other words, it's water-based. So the hydrophilic regions can face those directions, whereas the hydrophobic regions have to face the interior of the membrane. Again, like we said, there are proteins embedded inside of the membrane, and these proteins will also have hydrophilic and hydrophobic properties. So this is just a general overview of what the cell membrane actually looks like. Now let's break down that model, the fluid mosaic model. So let's discuss first what that word fluid means. Now typically in any textbook, this is the picture that you see of the cell membrane, which is kind of misleading in the fact that phospholipids are actually moving around. They're held together by the fact that there are hydrophobic areas and hydrophilic areas, but those are not very strong interactions. So the phospholipids can move laterally within the cell membrane. They never really flip-flop across, because that would mean that the hydrophilic regions would have to cross over in hydrophobic areas, which is kind of a no-no. But the phospholipids can move laterally, so they will move around and move places. Additionally, those proteins that are embedded in the cell membrane can move through the cell membrane as well. They typically move more slowly because they're a lot larger, and some of them will attach to the cytoskeleton and use the cytoskeleton kind of like a little railroad to move around. But others are also held in place by the cytoskeleton. But that first part of the word, fluid mosaic. The fluid part means that this is not a stagnant, non-moving structure. There are things that are able to move within the cell membrane. Now, some extra little hints or tidbits of information about the fluidity of the membrane. As temperature cools, we know that molecules move more slowly, so they slow down their movement. So membranes will actually move from being more fluid to more of a solid state, and that all has to do with temperature. Some membranes have more unsaturated fatty acids than they do saturated fatty acids. So the ones that are unsaturated are more fluid, right? Those are the ones that are bent, and the ones that are saturated are straight. Basically, straight ones, the phospholipids are able to pack tightly, and the bent ones, they're not able to pack as tightly as we would see with the straight ones. So unsaturated fatty acid chains typically are found in organisms that live in colder temperatures, so that their membranes don't pack up tightly like they would if they were made of saturated fatty acids. Another adaptation that some animals have is that they have the steroid cholesterol found in their cell membranes as well. And this goes back to something very similar that you saw with the unsaturated versus saturated fatty acids. When we have cholesterol kind of stuck in between the phospholipids, they are not able to pack as tightly, so they're not able to solidify in colder temperatures. So some animals that live in very, very cold environments have this almost temperature buffer when they have cholesterol in their cell membranes. So again, the first part of the fluid mosaic model, the word fluid, is that these membranes are not stagnant. They're not stuck in place. They are moving around. The components of the cell membrane are moving around. The second part of that model, the word mosaic, comes from the fact that a cell membrane is almost like a collage, and I'm sure you've seen a mosaic before. A mosaic is a picture made up of smaller pieces of things. So the membrane is kind of like a mosaic in the fact that it's made of phospholipids and proteins. So it's a mixture of different kinds of things. And the proteins are usually there and determine the majority of a membrane's specific function. Now, looking at a more complex picture of a membrane, this is actually what the cell membrane would look like. As you can see, we still have the phospholipid bilayer. We have the proteins attached to the outside or stuck in the middle. We also have things like the extracellular matrix attached to the proteins in the cell membrane. So the structure of the cell membrane is a lot more complicated than what we really give it credit for. And going a little bit more in depth with the types of proteins, you'll notice on this diagram there are proteins labeled peripheral proteins and there are proteins labeled integral proteins. Let's take a look at what those terms actually mean. Peripheral proteins are surface proteins. They're stuck to either the internal surface or the external surface of the cell membrane. Integral proteins go the whole way through the cell membrane. They are integrated. You can kind of think of it like that. And because they're going through the membrane, we call them transmembrane proteins, and they also, like we've talked about before, have the hydrophilic and hydrophobic portions. So the hydrophobic portions would be on the inside, and the hydrophilic portions would be on the outside. If we would take a look at what this, let's say for example, this protein blob would look like as its actual structure, this is what it would look like as an amino acid chain. So as you can see, the folds and the things that are nonpolar are on the inside, and the things that are polar or hydrophilic are found on the outside. Now, these proteins are extremely important to the functioning of cell membranes, and again, we'll take a look at each of these a little bit more in depth at some point within the year, but just kind of realize that these proteins do all kinds of different things within the cell membrane and can be specialized based on the type of cell we're looking at. For example, if we're talking about an immune cell, they're probably going to have a lot of proteins that are dealing with cell-to-cell recognition. They're going to be able to recognize viruses, foreign bacteria, foreign objects. Whereas something that is secreting something like that's in the pancreas or the liver, they're probably going to have a lot of proteins that are involved in transport. So let's look at each one of these different functions visually. Now, one thing we will be talking about a lot in the next upcoming chapters is cell transport. So we're talking about diffusion, osmosis, facilitated diffusion, active transport, all of those different ways that cells are able to move things into or out of themselves. So we'll look more at that definitely coming up. Another function that membrane proteins have is they are actually able to catalyze reactions, so they're able to be enzymes and perform reactions for the cytoplasm. After we finish up cell transport, we're going to do a chapter on signal transduction pathways. So what this is, just in a very quick nutshell, this type of receptor protein will attach a signal molecule, and then this process will set off a series of events to send a signal into the cell. So signal transduction is a way that cells can communicate with each other, one cell to the next. Another function, like we said with immune cells, cell-to-cell recognition. If we have a protein on cell A that has a carbohydrate attached to it, it can recognize and fit with another protein on cell B. So this cell is able to recognize this cell based on the fit, which is very similar to cell or example E. Back in chapter 6, we talked about different types of intercellular junctions, things like desmosomes and plasmodesmata and things like that. Proteins are involved in the joining. So those gap junctions, those tight junctions where one cell is actually attached to a neighboring cell. That's what we're talking about with intercellular junctions. And lastly, proteins can also function in attachment to the cytoskeleton or the extracellular matrix. So the proteins that are in and embedded inside the membrane can form structural components and attach to things that are outside of the cell and things that are inside of the cell that are there for cell structure and cell support. So just to kind of summarize today's quick lesson, the major points that I want you to kind of get is what is the structure of the membrane? So what's the component, what is made up of it, and what are the functions of that? So we will tomorrow in class build a model of a cell membrane. So be prepared to build a model that looks something like the simplified one, which I'm trying to find it way back here. Be prepared to build a model that looks something like this.